Bismillahirrahmanirrahim. Welcome to Biology Online class. This is Salmakta Sweetie, and in this video today I am going to discussing about biotechnology, and I am going to discussing details one of the biotechnology. That name is tissue culture. Let's know about biotechnology. The word biotechnology was derived from two words, that is bio meaning living things and technology meaning application of science for goods and services. So biotechnology is the application of science to the processing of living things for providing goods and services. That is, here we use living things, for example plants or animals or microorganisms, for making our products or goods, and for this making we apply scientific application to the living organisms, and the whole processing is called biotechnology. The word biotechnology was coined by Carl Ehrlich in 1919. So this is a very much new term, but we use biotechnological products from very ancient times. For example, we make curds from milk, and curd is a biotechnological product because here we use bacteria for making curd from the milk, and as bacteria is a living thing, so curd is a biotechnological product. According to Ericke, who coined the term, biotechnology was all lines of work by which products are produced from raw materials with the aid of living things. That is, we are using living things to make products, and making the products from living things, we use some works, and all lines of work related to the product making is commonly called biotechnology. Tissue culture is one of the multidimensional utilization of biotechnology, and as it is very important, so we have to know about tissue culture. Let's know about tissue culture. All of you have heard about tissue. Tissue is a group of cells or a cluster of cells that are originated from same cell, and the group of cells do the same function, and if cluster of cells do same function, and if they are originated from a same origin, then we can call the group of cell a tissue. And tissue culture is culturing a tissue. So culturing is what is culturing? Culturing is growing something in a nutrient medium. That is, nutrient medium is such like food, and if we give food, then anything will grow. And if we give food to the tissue, then it will grow, and this process is called tissue culture. So tissue culture is a technique of growing cells, tissues, organs, or other parts of living organism in a sterile environment on a nutrient medium. A sterile environment. What is a sterile environment? A sterile environment is a germ-free environment. That is, there is no germ and there is no chance to be attacked by the germs. Tissue culture had its origin at the beginning of the 20th century with the work of Gottlieb Haberland. He worked with plants, and this is his image. Though tissue culture is applied to both of the plants or animals or microorganisms, but plant tissue culture is most widely used. So let's know about the plant tissue culture basics. Modern plant tissue culture is performed under aseptic condition. What is aseptic condition? Aseptic condition is free of the conditions that is free of microorganisms. As the living plant in the environment are naturally contaminated. Contaminated, that means there is there is many microorganisms on the surfaces, and so surfaces must be germ free or free, and the making of germ free is called sterilization. So the plant tissue that we will use for next step of the tissue culture must be sterilized or must be made germ free, and for this we will use some chemical, alcohol, sodium or calcium hypochlorite. And in the plant tissue culture we use a special type of cells that is called totipotent cells. They have special characteristics, we will know in the next slide. And in plant tissue culture we will use agar in the medium, that is in the nutrient medium we will use agar. Agar is a polysaccharide powder derived from algae, and it make the medium solid. And another term will come that is callus. Callus is unorganized mass of cells. So let's know about totipotency. What is totipotency? Totipotency is the ability ability of a tissue or an organ or a plant part or any kind of cells that can give rise to a new plant. That is, we will take one cell or one tissue or one organ, and from that we can make a whole new plant. And who have this ability to become a new plant is called totipotent cell. And this is the baseline for the plant tissue culture, because we use the totipotent cell for making a new plant. And here in this picture you are seeing this is the root of a carrot plant, and we will separate some cells from this and we will give in the medium that will give it nourishment. And inside this a single cell will divide and further divide and give rise to a new plant. And so the ability of any cell to become a new plant is called totipotency. We'll use some growth hormones or growth regulators in the process, and growth hormones play an important role in growth and differentiation of cultured cell or tissues. We'll use such like auxin that regulate root growth, cytokinin that regulate shoot growths, gibberellin that regulate cell enlargement, and abscisic acids and ethylene are plant stress hormone. Tissue culture is a continuous process, and it is done in many steps. Let's know about the steps in tissue culture process. Step 1 is selection of a mother plant. We will select a mother plant from which we will make new plant, and during the selection we will select a high quality plant, that is a healthy and disease resistant plant. Here this is a healthy plant and this is a disease plant. By the appearance we can recognize them, and we will obviously select the healthy plant. The step 2 is preparation of culture medium. Culture medium is the nutrient, and culture medium are generally composed of inorganic salt plus a few organic nutrients, vitamins and plant hormones. Culture medium is prepared by mixing all the elements with distilled water, and sometimes we will add agar that will make the medium semi-solid. Here there is the whole constituents or whole the ingredients of the medium are in powder form, and here they are dissolved in water, and this is the final medium that has been made. The next step is the establishment of sterilized medium. After preparation of the culture medium, the medium is sterilized. What is sterilization? Sterilization is the making microorganisms or germ free. It is done by autoclave machine. Autoclave machine is a complex machine in which we can give a fixed temperature or pressure, and it will operate for a fixed time. And in this process we will use 121 degrees Celsius temperature, 15 pound pressure, and they will be applied for 20 minutes. And by this time the culture medium or nutrient medium will be germ free. That is, this temperature and pressure will kill all the microorganisms inside the medium. And then the medium is cooled and it will turn into semi-solid state. Explants. What is explant? Explant is the part of the plant that we will separate from the mother plant for the next step of the tissue culture. That is, the part of the plant that we will use for the tissue culture is called explant, and explants are then placed in the surface of the solid medium, and sometimes it will be placed into a liquid medium, that is called suspension culture. Here small part from the leaves is separated and it will be placed in the surface part of the medium. As there is various nutrients in the medium, it will the cells in the tissue will divide and it will give rise to a plantlet, that is baby plant, or a callus. What is callus? Callus is a cluster of cells without differentiation. That is, all of the cells will be same. There is no difference in the cells. And differentiation, what is differentiation? Differentiation is the process of being different cells from a cell. That is, when cell divides, the new cells will give rise to new organs, new parts of a plant. But in the callus, all of the cells are same. There is no difference in the cells. After that, from the callus, the cells will be transferred to a medium, and there will be development of shoots or roots. But if there is no roots in this medium, then a new medium will be made, and in which there will be root developing hormones, and the callus from here will be transferred to the new medium. Here there is high amount of root developing hormones, and it will facilitate the development of root. And by growing in this medium, both roots and shoots has been developed. And after that, the final step is the transfer to natural environment or field. That is, when plant with root and shoots are developed, then it is separated from the flask and they are hardened off. What is hardening off? That is adaptation period, and they are given to a environment that have lower humidity, and it will make them to adapt to the natural environment, and this adapting process is called hardening off. And when they are adapt, they are called matured plantlets. The matured plantlets are separated and the mediums are washed off, and then they are planted in the soil. So we are having many plants from a small part of a plant, and that's why the tissue culture is used. So in summary, we will know the steps of tissue culture. At first we will separate some part of the plant, and it will be placed on a liquid on a medium surface, and here it will grow or divide and give rise to callus. Callus is placed on another medium that facilitate the differentiation of cell. But if there is no root, then these will be transferred to a new medium, and that will facilitate the formation of roots. And after the shoot and root development, the plants are planted into the environment. So this is the overall process of tissue culture. This is all for today. Thank you all. Allah Hafiz.